We have studied mainly photosynthesis with reference to plants, but today's topic especially focus on bacteria. Plants are responsible for supplying foods to everything, that is okay. But what is the bacteria? Do they produce sufficient amount of carbohydrate that can be supporting the next levels of food chain? Our question answer may be no, but still they have got importance. They have importance, I would say, at least in two cases. First, as mentioned by madam, that during the process of evolution, it is said that cyanobacteria are the first organism which has originated on this earth. At the time of evolution, somewhere around 4.6 million years ago, the earth was evolved, and we have somewhere around, if I am not wrong, 3.5 billion year ago.
Yes.
We have fossil records. Fossil records says that cyanobacteria is the first living system which has originated on this earth. So cyanobacteria, during the oxygenic photosynthesis, they release oxygen as a product, and this oxygen was accumulated into the environment, and this oxygen containing environment was created, which is necessary for the growth of eukaryotic organisms. So this credit ultimately goes to this cyanobacteria. And second thing, the plants, we are giving maximum credit for photosynthesis to plants. We know that the organelle responsible for photosynthesis is a chloroplast. Chloroplast, we know that chloroplast, there are two organelles, one is chloroplast and another is mitochondria. These two organelles are having its own DNA. It is said that during the process of evolution, these cyanobacteria enter into another living system, and they grow as endosymbionts, and ultimately they lost their individual existence and they become organelle of this plant cell.
So that is how evolution adapted the new environment, which was at that time on the earth crust anaerobic environment, as well as afterwards converted to moisture and possibility of oxygen. So gradually they have started in a eukaryotic plants kingdom.
Yes, and by the time the maximum of the oxygen has been now added in our environment by plants, but the initiators, the founders, we have to believe that it was the cyanobacteria only. So this bacterial photosynthesis, the major difference in bacteria and plant is the light harvesting pigment. We know that chlorophyll A and chlorophyll B are the important pigments of plant photosynthesis. Similar kind of pigments are available in bacteria, but they are called bacteriochlorophyll. So here is a structure that gives you idea about chlorophyll A, which is a common photosynthetic pigment found in a plant. We can see the absorbance spectra. This is a chlorophyll A that has two peak somewhere around 400 and 700 nanometer. The additional peak we are observing, that is carotenoids. As madam has mentioned, there are two types of light harvesting pigments. One is a major pigment, another we can say it as accessory pigments, and carotenoids are one of the most important candidate coming under the category of accessory light harvesting pigment. So this is the structure and light absorption spectra of a chlorophyll A of a plant. Similarly, this is bacteriochlorophyll A. The major difference you will find over here is the number of peaks. In previous case there were only two peaks, but here we can see there is a peak somewhere around 400 nanometer wavelength light and 800, and one peak is extending beyond visible light.
Yes.
So that is around 900 or so. In addition to that, carotenoid is also again of different types other than plants in bacteria. So once we speak about the photosynthetic bacteria, we can classify by and large mainly them on the basis of how do they do photosynthesis. As we have mentioned earlier, photosynthesis in bacteria are of two types. One type use both photosystem, that is PS2 and PS1. When this Z form of cyclic system is used, it result in a photolysis of water. So obviously there is going to be the production of oxygen. So such photosynthetic bacteria are called oxygenic photosynthetic bacteria. Another group which operate only photosystem 1, which does not utilize hydrogen and oxygen combination, that is water, as an electron donor. They do not split the molecule of H2O, and so they do not produce oxygen. They operate only PS1, 700 nanometer wavelength light, so they do not produce oxygen, so their photosynthetic activity is called anoxygenic photosynthetic bacteria. Oxygenic photosynthetic bacteria, as we have discussed a lot about cyanobacteria, and other candidate in this is prochlorophytes, which has been recently identified. The another category, anoxygenic photosynthetic bacteria, has wide variety of organisms. Mainly again they are classified on the basis of the source of electron donor which they are using. Mainly they use hydrogen sulfide as their source of electron. So again here we have two groups. One group is utilizing hydrogen sulfide as electron donor, another group does not use hydrogen sulfide as electron donor, but instead of that they use some other organic molecules. So they are called organic electron donor utilizing organism, whereas the other one is H2S electron donor utilizing organism. Again, depending on the carotenoids and light harvesting pigment, the overall color of bacteria will be either green or purple. So depending on the pigments, there can be purple sulfur bacteria, green sulfur bacteria. On the other hand, purple non-sulfur bacteria. See here all the name explain itself, like purple non-sulfur means they will not utilize sulfur as electron donor. On the other hand, green sulfur bacteria can be also be green sulfur or green non-sulfur bacteria. This classification, in the very beginning also madam has mentioned that phylogenetic tree, and that tree has been synthesized mainly on the basis of 16S rRNA study. We know that Carl Hughes and his collaborator has done a very good job in classification of this organism. So this photosynthetic bacteria comes under eubacteria, they are not archaebacteria. As we have mentioned, oxygenic photosynthetic bacteria and anoxygenic photosynthetic bacteria, there are two types. Cyanobacteria and prochlorophytes comes under the category of oxygenic photosynthetic bacteria. Anoxygenic has previously discussed, there are wide variety: purple sulfur, non-sulfur, green sulfur bacteria, heliobacteria, and green gliding bacteria. So let us start now with the oxygenic photosynthetic bacteria. This occurs in cyanobacteria and prochlorophytes. The synthesis of carbohydrate, here both the light reaction and dark reaction, both the reactions will be operated. As a result of dark reaction, there will be the synthesis of carbohydrate. This result in the release of oxygen. As this are releasing oxygen, we can understand that this organism, oxygenic photosynthetic bacteria, utilizes both the photosystem, that is PS2 and PS1. At the same time, they will remove carbon dioxide from atmosphere. So PS1 and PS2, both the operating systems will be operated in cyanobacteria. This is a phylogenetic view where we have highlighted just cyanobacteria so that we can identify where is the location of this photosynthetic bacteria. As we are right now speaking about cyanobacteria, we have gathered some beautiful pictures, and in the first picture says it seems like it is eutrophication. So the presence of sometimes it happens like if any pond or river is having containing some polluted material that is rich in minerals and all that, so that supports the growth of microorganism. So in that case it is not always the algae that is growing, but sometimes cyanobacteria like bacteria also overgrow and they create such greenish water as we have seen just. And the next figure is bacteria, cyanobacteria. we may think like this is a hyphal structure, how come this can be a bacteria? But we know that the bacteria may arrange, they have different forms of arrangement, they may arrange in a chain like structure. So different cyanobacteria, these are two figures.
So sir, what you are saying is that what we saw on the rocks near the ocean, all these are actually not other things, but they are one kind of bacteria.
Yes.
And they have capacity to stick themselves on the rocks, on the stones, with their holdfast. And as you mentioned, there are some gliding bacteria.
Yes, gliding.
So these gliding bacteria are growing as if they are mycelial structure. Actually, they are not belonging to fungi. But they are looking like mycelial structure, and they, on the surface they overall spread. So they are named as gliding bacteria.
Yes. Because usually we have seen that microorganism can move in a liquid medium with the help of flagella. But it is a characteristics of some bacteria to move over the solid surface.
And also to make them adhere to the solid surface, like having holdfast.
Holdfast is a
So
structure.
it is very interesting to understand all these things. That how these bacteria are doing work also in light and dark. Also in presence and absence of oxygen, in both the conditions they are working.
Because they are a primitive kind of cell, so they have, God has given them almost all permission,
all capacities.
and capacities. So cyanobacteria, they are having, they can be categorized as a gram negative. I would like to tell dear friends that gram negative and gram positive is classification which is considered the basic classification in bacteria, that is on the basis of cell wall chemistry. So studying cell wall chemistry of cyanobacteria, they are found to be gram negative, and at present there are more than 1,000 species have been identified and reported. As we have seen, major light harvesting pigment in cyanobacteria is chlorophyll A. During this talk, wherever we will speak LHP, that stands for light harvesting pigment. So major light harvesting pigment in cyanobacteria is chlorophyll A, whereas other accessory pigments which are not, we can say they are not the part of reaction center, but at least they can channelize the electrons towards the reaction center, and they help in gathering as much as the photons possible. So these are called minor light harvesting pigments. The phycobiliproteins, phycocyanins, L-phycocyanin and carotenoids are the example of minor light harvesting pigments in cyanobacteria. Apart from this, as we have just discussed, as madam has said, that these prokaryotic systems has almost all capacity. So another additional feature they are having is they have additional light harvesting pigments. Some of the cyanobacteria have additional pigments, and these pigments example is phycoerythrin and phycoerythrocyanin. We know that erythrin related to red and erythrocyanin related to blue. So this different coloration of pigments give ultimate different coloration to the culture and overall colony of the bacteria. What is the, we have studied that in plants chloroplast is the organelle which is responsible for photosynthesis, but here bacteria is of comparable size to chloroplast, so it is not possible to have chloroplast as a organelle inside the bacteria. So how this bacteria manage, how do they overcome the problem of where to keep these light harvesting pigments? So the best what they have done is development of chlorosome. Now here we can say chloroplast and chlorosome, they have similar function, but the difference is chlorosomes are a structure which are not in connection to the cytoplasmic membrane. They are lie just adjacent to cytoplasmic membrane, and chlorosomes are having concentrated form of light harvesting pigments.
So sir, you mean to say that chlorosomes are the space where they put or they keep the room for the chloroplast kind of the pigment. So it is a separate room.
Yeah, kind of a separate room.
Okay, very interesting.
And they have a flattened sac of thylakoid that is containing chlorophyll A. Here the outer layer of thylakoid, sometimes it is having a droplet like structures, which is made up of phycobiliprotein. This is called phycobilisome. The presence of phycobilisome is unique to oxygenic photosynthetic bacteria. Such phycobilisomes are not observed in anoxygenic photosynthetic bacteria. We will take two examples of cyanobacteria with their special features, like oscillatorian cyanobacteria form filamentous structure, as you have mentioned very recently that though they are unicellular, but they form a filamentous structure, and it is also called trichome. The another is Nostoclean cyanobacteria. This is very interesting because we have given maximum credit to cyanobacteria for creating our environment and for being the first living system on this earth. Not only this, cyanobacteria have not stopped their work here only. They have done additional many things, and one example to this is nitrogen fixation. We know that it is the capacity of prokaryotes only to fix atmospheric nitrogen, and we know symbiotic nitrogen fixer like rhizobium and free living nitrogen fixer like azotobacter. But here cyanobacteria are also having a capacity to fix atmospheric nitrogen, and that is why cyanobacteria is one of the candidate which can be used as a fertilizer. So here we know that this cyanobacteria is a photosynthetic organism. They will produce, they are oxygenic photosynthetic organisms, so they will definitely produce oxygen. On the other hand, madam, this nitrogen fixation process is sensitive to oxygen.
Yes.
So the matter of interesting matter over here is how do they manage where one cell is producing oxygen and on the other cell they are having mechanism for fixation of nitrogen which is sensitive, and they have overcome this problem by the synthesis of a specialized structure called heterocyst. So as we can see in the figure, heterocysts are the bigger structures. As we are having this arrow over here, this is a heterocyst. Here again there is a heterocyst. There is a one more heterocyst. You will find that in a chain of a normal vegetative bacterial cell, there is a transformed cell in between. This gives a kind of a necklace kind of appearance. So here in this figure, this is the heterocyst. We know that nitrogen fixation is sensitive to oxygen because this process is carried out by